And we now turn to Dr. Reichardt, who's going to tell us about an update on liposarcoma. Thank you.
Yeah, Denise, thank you very much. Just as an introduction, a talk like this on a subtype, including subtypes of the subtype, would not have been possible a couple of years ago. So it's really due to a couple of new things going on, especially in liposarcoma. Time has been advanced a little bit, so I will probably need to skip a couple of slides, but just give you an overview what's currently going on, especially in liposarcoma. So mentioning liposarcoma, that is about 15 to 20% of all soft tissue sarcomas. That's a study 10 years ago from the French and Italian groups, and they looked at the distribution of several subtypes. And that was actually a very interesting thing because GIST has been thought to be a very rare disease, and it's actually the most frequent subtype of soft tissue sarcoma, followed by leiomyosarcoma and liposarcoma. Probably liposarcoma as a whole group is the second most frequent soft tissue sarcoma, at least in adults, and that is adults only. Well, if we say liposarcoma, that again is not one diagnosis, actually 4 diagnoses. Liposarcoma, the most frequent subtype of the subtype, is the atypical lipomatous tumor. That's what we think about a 0.5 grade tumor. You know the grading, it's grade 1, grade 2, grade 3, following aggressiveness. And the ALTs, they are not even grade 1, they are even less than grade 1. So it's still a malignant disease, but it's very, very low malignancy. It's actually the only sarcoma where an expected R1 resection is allowed and standard of care. So you can take out these tumors marginally. They may or can occasionally recur, but not always. So that's a special situation. We do put them in the group with the well-differentiated liposarcomas, and that is a little less than half of all liposarcomas. Most interesting from, well, actually current understanding is the dedifferentiated liposarcoma. That's 30 to 35%. And it's very important to know that the well-diff lipose and the dediff lipose, that's basically a family. So they coexist in virtually all cases. So in all dediff lipose, that is only part of the story. And in all dediff lipose, there are segments of well-differentiated lipose. So what we do normally see is a mixture of well-diff and dediff liposarcoma, and it's very important to understand. I will show you an impressive picture in a minute. An interesting subgroup, again, is the myxoid liposarcoma, and that's again a mix. We call it myxoid / round cell, because in the myxoid liposarcomas, there may be a proportion of round cells. That's the more aggressive tumor. So the more round cells, the more aggressive. If it's a myxoid without round cells, that is a very low aggressive liposarcoma. And if there is a lot of round cells, it's rather very aggressive. So that's again a mixture. And the histology does need to give information on the percentage of round cells to get an impression whether we are dealing with a very aggressive or a less aggressive tumor. However, it's not really well defined what the cutoff is. The cutoff still used to be 5%, but that's maybe inappropriate, as was just shown by the Italian sarcoma group during this year's ASCO, that 5 is maybe not an appropriate cutoff for defining aggressiveness. Maybe more round cells are needed to really call it a high-grade tumor here, but that's still under discussion. The rarest subtype is the pleomorphic liposarcoma. That is just 5%. So at this moment, liposarcoma in general, 15 to 20%, consists of 4 different subtypes of the subtype, and it goes down to about 5% of the 15%. So we are cutting down to very small numbers, putting them in even smaller drawers. So now we have a huge cupboard with big drawers, small drawers, and very tiny drawers that contain a single patient once a year in a country like even Germany or so. So it's getting increasingly differentiated, and we need to address all these tumors quite specifically, as Robin Jones already pointed out. This is what I just mentioned, the mixture of well-diff and D-diff liposarcoma. So if you, does this have a pointer? Okay, so that looks like a fried egg, so to say. That is a liposarcoma, and you can see it looks different. Here it's more pronounced. That is the liposarcoma over here, and it looks different. So if you do see such a picture and you take the biopsy from this part, you will get this, and that is a well-differentiated liposarcoma. Pathologists will say, well, nothing serious, well-diff lipo. Don't worry. If you take the biopsy here, the pathologist says, oh my God, that is a very aggressive grade 3 dedifferentiated liposarcoma. And that is the reason why, and later on today, Jens Jakob will give a presentation, image, board, biopsy, in this order. So in a sarcoma, soft tissue sarcoma, a biopsy should never be done before carefully analyzing the image, because you need to do the biopsy at the right side. And this is especially important in the liposarcoma story. So we do see these huge tumors, 5-10 kilograms in the abdomen, in the retroperitoneum, and it's always a mixture of well-diff and D-diff. There are complete well-differentiated tumors. Yes, they do not necessarily contain D-diff, but in a dedifferentiated liposarcoma, a big or a small proportion, but at least some, is always a well-differentiated. So if you take the wrong biopsy, you will get the wrong pathology report, either under or over estimating the aggressiveness. And that's the reason why for some of the ongoing studies, the follow-up of the patient is to be taken into consideration, because these tumors wearing biology, well-differentiated liposarcoma, we're talking about progress over years. So they grow constantly, but it can take 5 years for the next recurrence. In a D-diff aggressive liposarcoma, we're talking about weeks, as in other G3 sarcomas. So that's very important to keep in mind. Another thing is, in the retroperitoneal liposarcomas, as you can imagine, in the abdomen or in the retroperitoneum, there is a lot of fatty tissue. That's normal. Everybody in the room has that, more or less. And in these liposarcoma patients, all this fatty tissue is affected by malignant transformation. So even the normal fatty tissue does show MDM2 overexpression. That is characteristic for this liposarcoma. And all the normal fatty tissue has this transformation, and therefore it's even theoretically completely possible to resect these tumors in total. You would have to scratch all fatty cells from the abdominal cavity or the retroperitoneum out to take all the tissue out. That is absolutely impossible, and therefore these tumors virtually always recur after surgical procedure. Something very special for the liposarcoma subgroup. I will skip this. This has already been discussed by Robin. Just mentioning something. This is the study he mentioned from the Italian, Spanish, French, and Polish sarcoma groups on neoadjuvant chemotherapy, which did show superiority of the standard chemotherapy. By the way, the reason is not that the standard chemotherapy is superior, but the so-called histotype-tailored chemotherapy was very poorly designed. Actually, nobody would ever use any of these regimens in the first-line setting, by the way, so the result did not come unexpected, I would say. The only subtype where the histotype-tailored treatment did as well as the standard therapy were the myxoid liposarcomas, which is a subtype of disease where trabectedin is exceptionally active, and therefore this was, as you can see here, for all, yeah, that's the laser. For all subtypes, standard chemotherapy was better, with the exception of the myxoid lipose. There is equal activity for the standard chemotherapy and trabectedin. So this was extended, this comparison, and the results were shown during this year's ASCO. I will skip all this. That has been already mentioned by Robin Jones. That is something to support what I already said. A study by the EORTC, the European Sarcoma Study Group, the STRASS 1 study. Robin mentioned the STRASS 2 study. That's the STRASS 1 study looking into preoperative radiation followed by surgery or immediate surgery. So do these patients with a retroperitoneal tumor benefit from preoperative radiation therapy? Only patients with leiomyosarcoma or liposarcoma, these two subtypes, were included. That's the overall result, and you can see the preoperative radiotherapy does not offer any benefit. It's not better than immediate surgery. Looking into the subgroup, we do see that patients with liposarcoma seem to benefit from pre-op radiation therapy, whereas patients with leiomyosarcoma do not benefit. That is an extension of the study. Patients were included who had the same inclusion criteria as those put in the study, but could not be put in the study due to whatever reason. So they were also analyzed like this. That is the study cohort, and those are patients who could have been in the study but have not been put on the study. So the overall group is much bigger now. And we do see, interestingly, that patients with the low-grade liposarcomas, G1, G2, they do benefit from preoperative radiation therapy, whereas those with the G3 high-grade dedifferentiated liposarcoma do not benefit. So in total, this study shows that patients with a leiomyosarcoma and with a high-grade dedifferentiated liposarcoma do not benefit from pre-op radiation therapy. Why is that so? Very easy. These tumors tend to metastasize early on, so the general overall prognosis of these patients is not from the local situation, but from the metastatic spread, which is obviously not influenced by a pre-op radiation therapy. Metastatic disease is already present at that time point, and the radiation to the local site does not change that. As I mentioned, in the low G1, G2, the well-differentiated or slightly dedifferentiated liposarcomas, that is a local problem. I already mentioned not only the tumor carries this genetic abnormality, the MDM2 overexpression, but also the normal fatty tissue around. So here the issue is local. That's where the story goes on, the local problem. And here pre-op radiation works. That's what you would expect. The problem is local, so a local treatment like radiation, in addition, prior to surgery, improves outcome. When the problem is systemic, then a local treatment is not necessarily helpful in addition to the surgical removal. And here, radiation therapy does not work. And that's why the EORTC is now doing STRASS 2 using pre-op chemo for the high-grade tumors. So if you want to improve outcome here, you need to do chemotherapy. It was already discussed whether this trial is appropriate or not. From my perspective, clearly speaking, it's inappropriate. I would never, ever randomize a patient with a big, high-grade leiomyosarcoma or liposarcoma to a control arm, surgery only. That is, from my perspective, my personal perspective, not ethical. We do offer these patients preoperative chemotherapy in combination with local hypothermia as a standard of care. I would never randomize a patient in this study, so we don't take part in the STRASS 2. If people think it's appropriate, that's fine. I do not think it's appropriate. Okay. Trabectedin is very active in especially the myxoid liposarcoma that I've already mentioned. We'll skip this in the interest of time. Pazopanib has already been mentioned by Robin, is one of the active studies in the overall group of sarcomas, with the exception of liposarcoma. Pazopanib everywhere is not licensed for liposarcoma therapy for any subtype, with the exception of liposarcoma. Eribulin is a drug that has been studied in several subtypes and is licensed only in liposarcoma. So that is a drug only in liposarcoma, whereas Pazopanib drug in anything but liposarcoma. Two concepts, or a couple of concepts, two of special interest have been looked into specifically for liposarcoma, and that's targeting the CDK4 amplification. CDK stands for cyclin-dependent kinase. That's something that keeps the cell working on. And liposarcomas do have an amplification here. So if that may contribute to the disease, it's sort of logic to inhibit that. So if CDK4 drives the disease, I put a brake on, try to inhibit CDK4 to inhibit disease progression. A couple of studies have been looking into that. There was some activity seen. So this is ongoing research whether CDK4 inhibitors may be helpful in liposarcoma. That's a very recent study looking into different chemotherapy treatments, and for the well-differentiated, differentiated liposarcomas, and see overall outcome. And it seems that for the EDIF liposarcomas, doxorubicin ifosfamide seems to be the most active regimen. It's actually our standard of care. We do offer all these patients this combination therapy. Doxorubicin alone, that is the blue line, seems clearly inferior to that. That's a problem in the studies where this did not really show, because all these studies were mixtures of different subtypes. But looking at the different subtypes, we do see differences between single agent and combination chemotherapy. And that obviously also applies to the dedifferentiated lipos, where the combo treatment does seem superior to doxorubicin alone. Selinexor is an exportin-1 inhibitor, also a specific drug that was studied selectively in dedifferentiated liposarcoma. This was actually the first and biggest study for this subgroup that was done. It has been published this year. It took a while because the results were not as hoped for, I would say. You can see over here, there is some activity with this type of drug, but it's rather marginal. There is a minute difference in progression-free survival. There is no real difference in overall survival. So this drug was, we were more optimistic to see results here, but the outcome was not as hoped for. And I personally doubt that this will be followed any longer, given the current options that are much more promising. So it's unlikely that this type of treatment will play a major role in the future. This was just briefly mentioned by Robin Jones, this specific cell therapy for the synovial and the myxoid round cell liposarcomas. So in brief, we do use T cells from the patients. We take them out of the patient. Then they are genetically modified. During that progress, the cells are modified in the lab. and the patient is treated with an aggressive chemotherapy. And once the chemotherapy is done and the cells are modified in the lab, we return the cells to the patient who has been prepared, so to say, by the chemotherapy. And these cells now can detect the tumor cells in the patient and work. Robin Jones has mentioned that this works quite nicely in synovial. It also works a little less nice in myxoid liposarcoma. It's extremely time-consuming to do this whole process. It has a lot of requirements, and the number of patients treated currently and likely in the future is very low. So this is probably not a treatment that will be, well, of major impact on the overall population. It may be helpful in a specific small subset of patients, but probably not in a large scale. A few just hints from this year's ASCO a couple of weeks ago with perspective in liposarcoma. There was a study presented with a combination of lenvatinib, a multi-targeted tyrosine kinase inhibitor, with eribulin. You remember I mentioned eribulin is not licensed in liposarcoma. This study was done in lipo and leiomyosarcoma. And interestingly, there were a couple of responses in the leiomyosarcomas and the liposarcomas. The progression-free survival by histology, no real difference. In overall survival, it seems to work better in leiomyosarcoma, what we would expect, because eribulin does not really work nicely in the liposarcomas. So with respect to today's topic, liposarcoma, probably not something that will have a major impact. Now it starts to get interesting. That is the hot topic, I would say, in the field of liposarcoma. And that's the so-called MDM2 inhibitors. A number of them are currently under clinical investigation, and I want to specifically mention two because that's ongoing work. MDM2 stands for mouse double minute 2, a little strange name. And this works basically, no, I deleted the slide, so I'm willing to discuss that. Tumor cells, no, all cells contain something that is called p53. p53 is one of the major tumor suppressor genes. So that is something in the cell that is always looking for bad guys hanging around, causing tumor growth. And if p53 is active, we can call it the police. They suppress cancer, tumor suppression. p53 is a tumor suppressor. If the tumor suppressor does not work, tumors grow. That seems logic. So suppression works, no tumor. Suppression does not work, tumor grows up. MDM2 does stop p53. So basically MDM2 does suppress the tumor suppressor. So if you suppress that should suppress tumors. The result is that we do see tumor growth. And again, if we suppress what suppresses the tumor growth suppressor, now it gets complicated, then we might see activity. So easily speaking, p53 is very important to suppress tumor growth. MDM2 stops the activity of p53, so that is bad, because now the tumor can grow. And if we stop MDM2, then p53 works properly again. Now, why is this important? MDM2 overexpression is more or less a prerequisite for the diagnosis of well-differentiated / dedifferentiated liposarcoma. They have it all. In some studies, we are looking for MDM2 positive D. diff liposarcomas. That's a little bit strange because there is actually no MDM2 negative D. diff lipo. If MDM2 is negative, it's not a D. diff lipo. Anyway, that is the ideal disease to study MDM2 inhibitors, to restore proper working of P53 to suppress tumor generation. Okay, this means we are talking about MDM2 inhibitors in well-diff, D-diff liposarcoma. 2 drugs of the same family are currently in advanced clinical study. That is a drug from company Boehringer Ingelheim. Actually, Ingelheim is a couple of miles from here, very nearby. This has not yet a real name. It's called BI 907828, and it's currently under investigation. We'll skip that. I explained this. Just a brief addition, MDM2 amplification is typical for D. diff lipos, but not exclusively. A number of different tumors do have MDM2 amplification. So you can see more or less all types of malignant disease here that can have a degree of MDM2 overexpression, but it's very rare. The majority is seen in the sarcoma field, in the liposarcomas. So this drug, BI 907828, is a highly potent MDM2 inhibitor. It's been studied in a small proportion, and it has shown nice and prolonged responses in some patients. And it seems to have prolonged activity. The median or the estimated PFS in this early part of the study was around 8 months, so that sounds quite promising. That's efficacy in the well-differentiated lipo. Basically, all patients did show some degree of tumor shrinkage. Overall response, those with a shrinkage more than 30%, was nearly 30%. Again, lasting quite a while. And this study, or this drug, the phase 1 is currently ongoing, expansion, and there will be a randomized phase 3 study in first line, looking into doxorubicin alone, compared to doxorubicin plus this MDM2 inhibitor. So that's a study about to start, randomized phase 3, first line liposarcoma, doxo versus doxo plus BI 907828. That is the poster from this year's ASCO showing work in progress, mentioning this study. This study is called Brightline number 1, and this is conducted in, well, nicely around the world, US, Canada, many countries in Europe, China, Australia, and so on. The study is about to start in the very near future. The second drug, this is also from this year's ASCO, has already matured. That is the MANTRA study. That is using an MDM2 inhibitor that has already a nice name. Miladican, shown here. It's from a company with the name of Rain. Difficult name for a person like me who likes the sun, but anyway, it's a nice company, nice people. The study is somewhat different. It's looking into the activity. It's also an MDM2 inhibitor. So everything I already explained applies to this drug as well. It's an MDM2 inhibitor, P53 and all that, you remember. Here we are looking in a refractory disease situation, and the drug is used as a single agent. So the comparison is here. Standard so far for these patients is trabectedin as a single agent in second, third line, or even fourth line. And the comparator is this new agent. So it's a randomized study comparing standard of care, trabectedin, to this MDM2 inhibitor. Accrual of patient was quite rapid, fast, so it's nearly completed. We expect completion of the recruitment within the next 2, 3, maybe 4 weeks, and then we'll see what the results will be. The major toxicity of the drug is hematotoxicity, namely thrombocytopenia. It seems to be manageable by drug holiday and dose adaptation. So something we know from a number of oral agents, that you need to use them carefully. How much, how many days on, how many days off therapy, that is something that needs to be established in the future. However, having said that, major thrombocytopenia, that opens the question that will be very interesting, how such type of drug works in combination with chemotherapy. In the other study, in combination with doxorubicin, that is something we will see, we will learn in the future how it works or whether it works or not. So that's 2 ongoing studies, one nearly completed, in second, third, fourth line, prior therapy, in comparison to trabectedin, single agent, and the other MDM2 inhibitor will be studied first line, first line in combination with doxorubicin, and the control arm will be doxorubicin single agent, which is considered standard by many people, excluding myself. I would never consider doxorubicin single agent as appropriate. I never use it. We always use combination, but in the context of such a study, it's control arm, so we have to accept it, but outside a study, I would not accept it as standard of care. So that's ongoing work. We will see what we have already seen and heard and read about MDM2 inhibitors sounds quite promising. The concept seems logic, and we hope that the results of this and other studies and also future studies will show that it's really as active as we hope. I think that's about it, and I'll be happy to take any questions. Thank you.
That was fantastic, Peter. Can you hear me? Can you hear me?
Okay, now? Okay. So I just mentioned, just to repeat it, I don't know whether you have heard it. Hi, Robin. Robin and the colleagues. I just mentioned that we start, this is one of our main agenda to be involved as early as possible into the research process. And I already mentioned that we are in contact with Rain and also with Boehringer in a very early setting now for the last 6 months, and start the discussion with the companies to bring the patient's voice and patient's experience into the companies. That was the comment. So this shows our agenda to be involved as early as possible, not in the moment when the trial is recruiting or maybe when the drug is in the market, but much earlier to bring the experience into the companies. But my question goes directly to, you know, Peter, that we have had some troubles, for example, with the approval of pexidartinib. That was one of the issues here in Europe, where we advocated very strong in the direction to give these drugs in the hand of experts. And unfortunately, EMA didn't follow our recommendation. So the question is, when I look to these kind of drugs now coming up, and also with the complexity of this disease, you talked about biopsy, and that we need knowledgeable pathologists, and we need a tumor board, and all these issues. We are moving more and more in the direction that these kind of diseases can be only handled by sarcoma centers. And so the question is, shouldn't we move more active in the direction of EMA, that these kinds of drugs really should given in the hands of experts and not just to the regular oncologists?
Definitely, I couldn't agree more. I mean, with pexidartinib, it's a special situation. The disease for which this drug is licensed in the US is not a malignant disease. It's a benign disease that can cause a lot of trouble to the patient, but it's not a malignant disease. And the European perspective is that it's unacceptable to offer a drug that may even kill a patient, that can have severe side effects, if it's not a malignant disease. The position from the US is somewhat different, so they license the drug, but in Europe they think, well, you cannot risk to kill a patient that does not have cancer with a drug. I mean, there are a lot of drugs around that can kill patients that are used for hypertension, diabetes, heart insufficiency, and so on. So the logic is not really 100%, but nevertheless, that is the issue. You are absolutely right. We do know from many drugs that in the wrong hands, they can cause a lot of problems, a lot of troubles that will not help the patients. So actually, it's in the vital interest of patients and therefore patient organizations to make sure that these difficult drugs are only used in experienced hands. If you want to drive a truck, a big truck, a bus, you need a special license. With the driver's license probably most of us have, we are allowed to drive a car, a small bicycle, but not a big truck, and never ever a bus with 50 people in. For every other bicycle type or car type, you need a special license. For all these drugs, you need one single license, that if you have a license and you can drive everything from a small car to the biggest available truck ever, every type of motor bicycle, everything, that's unimaginable. But in medicine, unfortunately, that is the case. Everybody can use a prescription and write something on. They don't even know what they write. They write a name, they check, oh, I need to spell, and they write something. They don't know what they are doing, but they are allowed to do. And the problem is for the patient, because they will either have severe problems with severe, dangerous, even life-threatening side effects, or they will be dosed in a way that will never ever help, or the treatment will be stopped. Ooh, too dangerous, stop it, and things like that. So that is something of the utmost importance, and I can only very much support anything in the direction of getting this type of drug only into experienced hands. And I'm sure Robin will share this position completely.
Yeah, no, I totally agree, Peter. I think that's a really, really important point. I think the other thing to emphasize is that recruitment has been so good into a lot of these trials, as you mentioned, particularly the MANTRA trial, where it's just been so rapid. And as you said, the trial is likely to close to enrollment over the next few weeks. So it's really, really good that we can do these randomized trials in specific patient groups. And I think, that's a really sort of positive sign to the community, because I think the concern previously had been, well, it's always challenging to do trials in sarcomas. It's difficult to do randomized trials in specific sarcoma subtypes, but that's clearly not the case, that we can do trials very efficiently and can enroll very, very, very quickly.
Okay, thank you very much. Again, Andrea from Italy. I'm working in the aeronautical sector, so I have nothing to do with the medical sector. But in the aeronautical sector, we have the so-called mutual recognition. Is it possible to push for EMA, because we have the clear example of desmoid, who is approved in the United States and is not approved yet in Europe. And what we should maybe do is push to EMA for a sort of mutual recognition for specialized cancer centers.
Yeah.
There have been some patient advocates in the room. We really emphasize that this should be the case, that EMA should license this drug, pexidartinib, really to selected centers, but they would be not willing to follow because they don't understand the whole topic. I think we need to move forward to have conversations with this, and the possibility is there because there's a strong relationship between Rare Cancers Europe, what we already have funded with Paolo Casali and all these guys, in moving to EMA and discuss this topic closer with EMA. You are absolutely right. We need to put these drugs in the right hands. Otherwise, it's a huge problem.
Yeah, because it shouldn't be difficult to paste and copy a mutual recognition principle from the aeronautical sector to the medical sector in this specific case.
Yeah. I mean, why would you not make a drug available? Because in unexperienced hands, it may be difficult to handle. That's like you would allow no big trucks, no buses, because not everybody can drive them. Say, well, we forbid drugs and trucks because a young person in his first year of driver's license cannot drive this. So we forbid these cars types. That's crazy. So if you need a special experience to use something, then give it only to those who do have the experience. And that applies for these drugs. If the drugs are exceptionally expensive, difficult to handle, dangerous, or only work in a very specific subgroup you need to understand and to identify and all that, then make it available to these experts and not to everybody instead of not making available to anybody. That is the wrong way. So I don't know how we can get there, but we should, of course, try to get there, and the regulators are the people to talk to.
Just to say as well, Andrea, that the patient voice in this is really, really important, particularly with the regulators. So your support is really, really valuable.
We can't hear anything.
Yeah, yes, okay. So it's not just a question about the safety or harming the patient, it's also about the question of outcome. Because we know when we send these patients, specifically our experience in GIST, when we send these patients to Peter or Robin or the other GIST experts, we know exactly that the outcome of the patient is much better. That means the patient lives longer because these guys know exactly what they are doing by managing the therapy. I think that's the big difference. So we are talking about life expectancy and not just by One part is to harm the patient and maybe do things wrong, but the other is to get the best out of the treatment if we send these patients to the experts. That's the difference.
I think we have a couple questions online. Roger, you had a question? I thought there was one more.
It wasn't a question. I wanted to follow up this discussion about specialist centres, which Peter, Marcus, and Robin have all had. We've got to remember within Europe that healthcare issues are national responsibilities. The regulator operates according to the rules set by the European Union. And the European Union gives the regulator the right to authorize the drug for marketing, but no other authority. The authority about how a drug is used within a nation is down to the individual nation. When we, as the UK, were in the EU, we took all advantage of that independence, and we created our own specialist networks, in around 2006, 2007. France did something very similar. Spain has followed. Germany has now followed. The Netherlands followed. And it's down to those healthcare systems to regulate how and where the drug is used, because that is European law.
Very good. Well, I think that will be the last word that we will have, Roger. Thank you so much. I think that's such an important call to action that we really need to be there. I see Mitch had a question. Is there a compassionate use regulation? Is that within Europe? Is that
It varies from country to country.
Country to country, so it's not across the.
No, very briefly, it varies between countries, it varies between companies, it varies between type of drugs, about the already available knowledge, the time we expect until the drug will be marketed and all that. So it's very complex. There is not an easy answer. For some of these agents, there is no compassionate use. For example, for tazemetostat, there is no compassionate use program, so we cannot get the drug, even for patients where it's licensed for in the US, so it's very difficult. It's interestingly licensed in Europe for a specific time of lymphoma. Okay. So there is a backdoor that may be used, but it's very difficult and a little bit dangerous and expensive to use, but there is a sort of a backdoor. But compassionate use is complex. I don't think we can discuss this in length. Just briefly, it differs from country to country, from drug to drug, from company to company. So
it's clearly not the answer for everything.
No easy single answer.
Yeah. So this has been just a fascinating discussion, Dr. Reichardt, Professor Jones. I think we see how important clinical research is and the role that the patients can have in bringing our voice to these important trials, whether it's helping to advocate for them, to get them done quickly so that questions are answered, to provide input, and then work with regulators as well. That clearly there is much work to be done. So thank you, Dr. Reichardt, Professor Jones, for being here.